in the scientific press in 1907. But of course, that then set a stage for a series of advances in understanding the molecular basis of inheritance. By the midpoint of the last century, of course, was the discovery of DNA's double helical structure, which was key in unraveling how it was that DNA was the information molecule essential for inheritance. And then advances in the 1960s, the 1970s, and the 1980s led to the creation of a new field of genomics, 80 years after the creation of the field of genetics. And in fact, shown here is the first time the word genomics was used in the scientific literature in a lead editorial of a new journal called Genomics. Now, the reason that a field was named called genomics in the late 1980s, and the reason there was a lot of excitement was because by the late 1980s, there was advanced planning for a once-in-a-kind audacious project that aimed to read out the sequence of all the DNA in humans. That is, of course, the genome. So reading out the sequence of the human genome. That project, of course, was known as the Human Genome Project, which began in 1990. I was very fortunate, as you heard in the introduction. I was training at Washington University and as a postdoctoral fellow and as a resident in pathology, and got to participate in the Human Genome Project from the very beginning. It was a big, audacious international project, unusual in scope and scale. It absolutely required thousands of very, very talented people from all around the world to pull it together. But at the end of the day, over the 13 years, it accomplished all of its goals, and it produced the first sequence of the human genome. Now, I think it's important to point out that the Human Genome Project, which really was a remarkable success that was guided by a series of publications that described how to accomplish the goals of the Human Genome Project, was, very much one of these things that 21 years ago, when it ended, it was as much a finish line as it was a starting line. And in many ways, I think of the end of the Human Genome Project, the finish line, is really just the start of everything I'm going to describe to you now. Now, I work at the National Institutes of Health in the United States. Our involvement in the Human Genome Project, and we were the institute created by the US Congress to lead the US's effort in the Human Genome Project, was all about thinking about health applications of genomics. There are many other applications of genomics that have emerged over the last 21 years, but my focus is going to be on using genomics to improve the practice of medicine. In fact, that was one of the reasons given for why we wanted to sequence the human genome during the Human Genome Project, because we thought we could better practice medicine if we could access information about each individual patient's genome and use that information as part of their clinical care. It therefore should perhaps not come as any surprise that during when the Human Genome Project ended, that whether you look at the popular press or the scientific press, you would end up seeing that the words genomic and medicine were being juxtaposed together. And this juxtaposing of genomics and medicine led to this phrase, genomic medicine. And that began being discussed about 21 years ago. The only problem with this is that while we could spell genomic medicine, it was a very, very blurry concept. We really didn't know what genomic medicine was going to look like, and we needed to bring it into focus. Now, we could define this idea, and we recognized that this was going to be an emerging medical discipline that involves using genomic information for an individual as part of their clinical care. And that sounded very reasonable, recognizing that each of us is unique, and some of our uniqueness is differences in our DNA, and some of our uniqueness is our physical and social environment. But we were poised with the sequence of a human genome in hand to now take a careful look at the DNA differences that exist amongst us and how that information might be useful for tailoring medical care of people. Now, there's other terms that are sometimes used for this, other phrases: personalized medicine, individualized medicine, precision medicine. I will tend to use genomic medicine because I'm going to stay focused on genomic information, whereas these other phrases and terms tend to broaden it even beyond genomics. So, what was that pivot to genomic medicine going to look like? What was it going to be like to move from that start finish line of the Human Genome Project to create it into a starting line that would eventually yield the realization of genomic medicine? Well, that pivot was going to involve the start of a new journey. And like the previous journey of the Human Genome Project, it was going to involve a series of steps. We don't even know what all those steps are going to be or were going to be. But what we do know is just like the Genome Project, we could take one step at a time and we could accomplish that. And then we could cross it off our list and then we could just keep going. I think we also appreciated that, unlike the Human Genome Project, or maybe like the Human Genome Project, this was not going to be a sprint. It was not going to involve one lab, one country, one scientist. It was going to be much more like a marathon. Many, many scientists all around the world running shoulder to shoulder for what inevitably take a longer time than the Human Genome Project, and recognizing that we were going to need to really rely on our endurance to eventually realize genomic medicine around the world. Now, what has transpired over the last 21 years has been a series of additional rounds of scientists thinking about what needed to be done to accomplish the goals as we pursued this journey en route to genomic medicine. And these sort of defined several eras. The most recent era is one that started at the beginning of the decade. At our institute, we publish strategic visions. You may be familiar with these. I show you the three. One that was published the day the Human Genome Project ended, another one in 2011, and then the most recent one in 2020, which I will come back and talk about at the end of my talk. But what I really want to emphasize here is these strategic visions provided guides for this journey from the Human Genome Project to the realization of genomic medicine. And those guides led to successes. And so, in a very rapid fashion, let me tell you what I think have been the incredible successes en route to genomic medicine. The first one is what the Genome Project did. The Genome Project, sequenced the human genome for the very first time and delivered the roughly 3 billion letters in order to the world that all of us now access through the internet and use as a framework for doing all sorts of research. But since the end of the Human Genome Project, there have been some spectacular advances that have moved us in closer and closer to realizing genomic medicine. First of all, we've reduced the cost of sequencing a human genome by more than 1,000,000-fold. There are all new sequencing technologies that have largely replaced what was done during the Human Genome Project. And as a result of that, we can now sequence a human genome for less than $1,000 US. And so we can cross that off our list, feeling very accomplished at these technological innovations. We've gone on and have used those inexpensive sequencing methods to now sequence millions of human genomes, and based on that, have cataloged literally hundreds of thousands of places in the human genome where we know that people vary. We have an incredibly rich catalog of variants, places and different spellings in our genome compared to other people. And that information is serving as the basis for then understanding how genomic variation might influence biological function. That is being augmented by an increasing number of advances in understanding how the human genome actually works. Let me remind you, the Genome Project delivered the sequence of the human genome, but it didn't deliver an interpretation of that sequence. But in the last 21 years, we have used an amazing amount of human creativity and innovation to figure out where are all the genes, increasingly, where are all the regulatory elements, where are all the parts of DNA that gets made into RNA and so forth. So while we have a significant amount more to learn about the function of the 3 billion letters of the human genome, I think we can say we know a lot more now in the 21 years since the end of the Human Genome Project. We've used these sequencing technologies, this information about genomic variation, increasing understanding of genome function, to now also move into human genetics and really begin to significantly advance our understanding about the genomic basis of human disease. Our greatest accomplishments have come in the arena of rare genetic diseases, where we now know the genomic basis for nearly 6,000 rare diseases, Whereas when the Genome Project started, we only knew the gene that when mutated caused 61 rare diseases. So from 1990 to now, we've gone from 61 rare diseases for which we understood the genomic basis to 6,000 or nearly 6,000 now. That has been remarkable. Meanwhile, we have made significant strides through the era of genome-wide association studies of trying to understand where are the regions of the human genome that contain variants that confer risk for more common diseases like hypertension, cardiovascular disease, Alzheimer's, autism, and so forth, and diabetes, and et cetera. Now, we still have a long way to go, but I think what we have seen with rare genetic diseases and setting up a circumstance for better understanding of more common genetic diseases, we really should look and say, wow, we've made significant strides. What has been particularly satisfying to me as a physician scientist, and what I really wanted to emphasize in my talk, was the fact that by the middle of last decade, things really started to come together with respect to efforts to start to use genomics in medicine. I mean, we had cheap DNA sequencing, these catalogs of genomic variants, better understanding of the functional landscape of the human genome, and we now had figured out the genomic basis of thousands of rare diseases and increasingly gaining insights about other diseases. So these developments really culminated in some of the earliest examples of genomic medicine being implemented. And so I just wanted to share with you what these examples are, because I think they really represent the beginning of us crossing that finish line where we are starting to realize genomic medicine. So you may ask me, what is the basis for these advances? And it really is the fact that with better and better knowledge of our variants that exist throughout all of our genomes, we have now gotten to the point where we can take a patient and for less than $1,000, we can generate their sequence. We can compare their patient's sequence to the reference genome sequence like that produced by the Human Genome Project, and we get a list of genomic variants. And when we look at those variants, we can interpret them and begin to implement genomic medicine. Now, remember this slide, because this slide I will come back to in a little while to make a very important point. It's a very nice slide, but I will admit now it is very oversimplified, but it sets up the next area I want to talk about, which are the actual examples where genomic medicine is absolutely here and now. You may ask me, what are those areas that I'm so excited about? Well, each of these could be a talk in and of themselves, but I wanted to at least share with you in a highlight form what I think are the exciting areas where genomic medicine has been implemented. The first is cancer. Cancer is a disease of the genome, and we can apply all these technologies for studying a normal genome to study a genome in a tumor and look at a cancer genome. This has been done in research substantially over the last 10 years, but increasingly is being used as part of clinical care. Patients who have cancer are increasingly having their cancer genome sequenced and analyzed, and with that information, it is changing the way they are treated. I believe that cancer is going to be the area of medicine most advanced because of genomics. In addition to cancer, it is now routine to be able to diagnose rare genetic diseases by sequencing a patient's genome. This is being done tens of thousands of times every single year in across the world. It's maybe even approaching hundreds of thousands of times across the world. In many settings, this is being done as part of a diagnostic workup of a young child or even an adult with an undiagnosed condition. But it's also being applied increasingly in acute settings, such as neonatal intensive care units, where there is now the ability in many countries to be able to sequence a newborn's genome, a newborn that is acutely ill, for which they don't know what is wrong with that child, and they can get a genome sequence in less than 24 hours. And that has been miraculous. It has saved lives because diagnoses have been made that previously would have never been made without genomic data about that acutely ill newborn. And with that, as a result, many neonatal intensive care units have the ability to send out and get genome sequences of their patients in less than 24 hours. The number one genomic test in the world today is a test that is called non-invasive prenatal genomic testing. It is a technology that basically draws a little bit of blood from a pregnant individual and detects the small amount of fetal DNA that floats around in that pregnant individual's bloodstream that comes from the baby. And with that as a screening method, you can look for aneuploidy, abnormal numbers of chromosomes. It is estimated that 6 to 8 million pregnant individuals will get this non-invasive prenatal test done. It has been widespread adopted around the world. And because of it, it has basically largely eliminated the use of things like amniocentesis for prenatal testing because you could use non-invasive methods to be able to access this. Again, it's utilized these new sequencing technologies. It has been remarkable how the uptake of this non-invasive prenatal genetic test is. Pharmacogenomics is certainly catching on, not as fast as some of these other areas, but we all know we differ in how we respond to medication. Increasingly, we are learning that the basis for that difference is often DNA variants that exist in genes that are part of drug metabolism pathways. And we can use that information to not design new drugs, but just to select the best drug for each patient. There are, many, many different types of hypertensive medications, asthma medications, psychiatric medications, and often it's just hit or miss. But pharmacogenomics is offering us the ability to pick the right drug for the right patient at the right dose using genomic information. The last area which is getting a lot of attention is genomics-based prevention. Increasingly now, especially for rare diseases that are known to be in families, you can find out in advance whether or not somebody is susceptible to colon cancer or to breast cancer or to some nutritional defect, as is often done in the case of young individuals, young children. And based on that, genomic information can be used to prevent advanced cancer by higher surveillance or changing a child's diet as necessary to be able to avoid any cognitive deficits resulting from having the wrong food being given to them because of some genetic defect. So the bottom line is genomic medicine is here and now. These 5 examples are just some of the earliest examples, but they're highly illustrative because they are telling us how genomic medicine is what it's actually going to look like. And so I will add this to my highlight reel, feeling very, very satisfied that we now see vivid examples of genomic medicine emerging. So that's the optimistic, incredibly wonderful highlights that I could tell you from the last few decades of genomics. I want to immediately point out that this has been a research journey and it has not been linear. And throughout the Human Genome Project and the 21 years since the Genome Project, there's lots of twists and turns, but in an abbreviated form, I've been able to give you the story. But I hope you realize it has not been a linear story. It has been complicated everywhere along the way. And like it has been a complicated story along the way, it remains a complicated story. Challenges always are right in front of us in science, and genomics is no exception. We face constant challenges during the Human Genome Project. We face constant challenges in the first decade, and now the second decade, and the third decade after the Human Genome Project. And so, knowing this is a bioinformatics conference, I was thinking about what challenges would you may be interested in, especially from a computational side of things. And so I decided to emphasize one particular challenge that we are now really addressing with respect to genomic medicine implementation. And it relates to this idea that I was a little too candid, or maybe I wasn't candid, maybe I was a little too magical when I described how we just sequence a patient's genome and instantly know what to do about that. The truth of the matter is, most of the time when we go to sequence a patient's genome, we sequence their genome, and when we go to round on that patient the next morning, we get a list of all those variants, and we don't know what most of those variants mean. It is still a massive challenge to interpret a sequence of a patient. Getting the data is easy, but and I showed you this slide, this was an intentional oversimplification. Going from a patient's genome sequence to implementing genomic medicine is not a solved problem. We have solved it in a few areas. We are making better and better progress every day. But this particular slide, going from a genome sequence to getting the variants to implementing genomic medicine is still a massive challenge that needs to be refined and improved upon. And it will involve a lot of computational experts around the world to help us do it. So I want to spend some time explaining this because I think it's a very interesting, and in many cases, people may not fully realize some of the things going on now to make this challenge a little less onerous. In order to do that, let me remind you, there are three major steps associated with analyzing a person's genome sequence. And I'm going to tell you about each of these three, and it will explain some of the things you may have read about going on now in human genomics. The first step of analyzing a person or a patient's genome sequence is you need to detect all the genomic variants in their generated genome sequence. So you need to have a patient, you sequence their genome, and you want to detect all the variants. In order to do that, you need to have a reference sequence. And we're going to talk later about pangenomes, and I list some of the common ones that are used. Some of you may know which ones they are. But let me unpack this a little bit more, because I want to make sure you realize what is a reference genome, what is a routine genome that we get from a patient, how do you generate it nowadays, and how do you generate it cheaply? So let me just make sure you're all clear. A reference genome with its little certification check mark and ribbon in the upper left is a very high quality representation of a genome. It is meticulously generated. It is generated using multiple DNA sequencing technologies, not just one. It has very little, if any, missing sequence. So it has complete sequence or nearly complete. And it costs today about 10,000 US dollars to generate through experts a reference genome sequence. Okay, so that's a reference sequence. That is not what you generate with a patient. What you generate with a patient is a routine genome sequence. A routine genome sequence is the purpose of it is to identify genomic variants. It's not to be a high quality representation. It is a diagnostic or a research tool. It is generated in a high throughput fashion, and a single sequencing technology is typically used. Just one, oftentimes Illumina-like short read technologies. It always has missing sequence. It is not about getting all the sequence because it's very inexpensive. This is what costs less than $1,000. When we talk about $1,000 genome or a routine genome sequence or a diagnostic sequence, we are talking about it being very inexpensive, less than $1,000. Okay, how do we generate a routine genome sequence, and then how do we use a reference? Well, the way we generate a routine genome sequence is we get DNA from somebody and we shear it all up and we analyze those little fragments using one of several technologies in a black box-like way, and we generate hundreds and thousands, if not millions, of generated sequence reads, oftentimes short sequence reads. We've got lots of them. Now, how do we stitch those reads together? Well, unlike the Human Genome Project, where we had to put them all together on our own in a de novo way, that is not the way we sequence genomes today routinely. We use a reference sequence to help assemble or really help generate that sequence. So how does that work? Well, you take these generated DNA sequence reads, and then you use a reference genome, like the genome produced by the Human Genome Project or ones that are available. But it's a high quality, nearly complete sequence representation of the human genome sequence. And then what you do is you use that reference to figure out and place all the reads that you have generated from your patient or from your research subject. So you take a read one at a time computationally, and you take it and you look for where it matches in the reference sequence. And where you find a place it matches, you say, okay, it's right there. Okay, good. I'm going to move it. I'm going to now have that represented in the routine genome sequence. And then you're going to have to do this a bunch more times with all the rest of the reads. So now you take a second read and you look to see where does it match in the reference genome. When you find it, boom, it goes over there. And then you take a third read, and the third read will find its place in the reference genome. And when you find it, you move it into position in the routine genome sequence. And you just keep doing this. But notice that when you grab sequence read number 4, it goes to try to find a position within the reference sequence, but it doesn't find one. So it can't be incorporated into the routine genome sequence. It goes in the trash bin, or it goes into a pile of non-matching sequences. So that's what you just keep doing. And as you can see, You're just going to start pulling these one read after another, and you're doing this literally hundreds of thousands of times as needed, so that you're able to generate lots and lots of data the best you can. And when a read doesn't match, it goes into the non-matching bin. So you are using the reference to assist in the generation of the sequence. Now, the other important point is by the time you're done with this, you have literally each line of that routine sequence has been read and localized many times. In fact, it's typically done at least 30 times or more, so that when you have placed a read in a given position, say 30 times, it's going to be pretty darn accurate. Okay? So what you have is highly accurate. At the same time, then you have your sequence of each, and now you can compare them. You've assembled or you've put together your routine genome sequence, you've put together your reference genome sequence, and now you compare them. And when you compare them, you are able to basically have an indication of what's different from one another. You compare to find differences. And when you find those differences, you find single letter differences, you find longer differences, you find deletions, insertions, you find all sorts of differences, is shown as yellow versus red in this particular slide. And from that, those variants, you could make a report of all the differences that exist in the patient's genome versus the reference genome. So that is how we generate a routine genome sequence of a patient or of a research participant inexpensively. However, keep in mind, when you do this typically with one reference genome. That one reference genome came from one person. And yet we all know that people differ. And so what is happening now in genomics is that we know that since that reference genome is critically important, it needs to be pretty well matched to the individual that you are sequencing. We are generating more and more, literally hundreds of reference genome sequences from a whole bunch of different people. You don't want to just work with one reference genome. Now, why is that? Well, let me explain why it's so important that you don't just work with one reference genome. Because if you work with one reference genome, when you go to put together the sequence routinely of an individual, you'll take all those reads, and as I just showed you on the previous slides, you'll get some that don't match, and then you put your sequence together. But with different references, you get different numbers of non-matching sequences. And you could just sort of see here, hypothetically, how each of these different reference sequences yield slightly different numbers of non-matching sequences. Look at number 5. It has a huge number of non-matching sequences. These are perfectly fine sequences, but because they didn't exist in reference 5, they don't get put into the routine genome sequence. Whereas reference sequences 1 and 6 are pretty darn good, and they're certainly better than, let's say, 2 or 5, or 7 is awful. And 8 is probably not so great either. So different reference genomes differ based on who you're sequencing and how many non-matching reads there are. This is what has led to the notion of a pangenome. A pangenome is basically an aggregated collection of reference genomes. It's basically a composite of multiple human reference genome sequences that together capture the breadth of human genomic variation, much better than any one human reference genome sequence. And because of that, it enables more accurate and complete detection of genomic variants across diverse populations. That is what a pangenome is all about. And the reason this is so important is, again, the idea, and this is all an active area of research, is that we could take the generated sequence reads from that patient, we could put it across the reference genome, and in doing so, by some fancy computational way, we would have very few non-matching sequences. We would end up with a more complete routine genome sequence. And so to summarize, and I hope you appreciate, this is an active area of research that we are trying to get away from using single genome references when we sequence a patient's genome, because when we do it, we end up with a bunch of non-matching reads. And yes, we detect a lot of variants, but ultimately, we want to use a pangenome, which will yield a sequence with many fewer non-matching reads. And so that at the end of the day, when we're trying to detect genomic variants, we will have, with a single reference, you have fewer true variants and more false variants. In contrast with a pangenome, you have more true variants and fewer false variants. This is why my institute, the National Human Genome Research Institute, has something called the Human Genome Reference Program. It is what is producing the human pangenome, featured on the cover of Nature magazine last year. And we are in the process of generating a human pangenome that will consist of hundreds of high-quality reference human genome sequences. So that is why I wanted to explain to you this very first step and why a pangenome and reference genome sequences are so important to detect all the genomic variants in the generated genome sequence. The second step, which will take shorter for me to describe, is the step where, okay, now you have a very good collection of variants, but now you need to filter and prioritize all the variants to figure out which are the ones that are likely clinically relevant, which are the ones that are pathologic in the case of rare genetic diseases. You're looking for a variant that is pathologic and causes that rare condition. And for this, you need a reference population database where you've aggregated genomic variants from many, many, many people. So let me unpack this just to make it very clear, is that the frequencies of genomic variants as diagnostics is that if we just think about rare diseases, because they're sort of the prototype here, the vast majority of the pathogenic, in other words, the causative genomic variants, the vast majority of them are rare. And so, but being rare doesn't mean that a genomic variant is pathogenic. It just means that it's one of the criteria. But being common most of the time means that a genomic variant is unlikely to be pathogenic. If it's frequent in the population, it is unlikely to cause a rare disease because rare diseases are rare. Therefore, genomic variants are initially filtered into groups that are common, you remove those, and then you keep behind those that are rare, because those are going to get prioritized attention. And then following filtering, the prioritized variants are further assessed for possible pathogenicity. Let me graphically show you what this means. You sequence a patient's genome, any one of your genomes we could sequence. When we did that, we would likely find 3 to 5 million variants in your genome compared to any reference or any series of references. Way too many variants to possibly be clinically relevant. But through this series of filtering steps and prioritizing steps, we sequentially keep reducing it down so that at the end of the day, we're probably looking at 10 to 20 variants that might play a role, for example, in a rare genetic disease. And that filtering process requires the presence of databases of variants from different world populations. And again, to put a fine point on this, the frequencies of genomic variants vary among ancestral populations. If we just looked at one population, we would get misled. Let me show you hypothetical examples. So here are 4 ancestral populations. Let's say population 1 is Europeans. Let's say population 2 are people from India. Population 3, maybe people from the Far East, et cetera, et cetera. If we find a variant like variant 1 that's relatively common in all populations, we throw it away. We filter it out, unlikely to be the cause of a rare disease. Variant 2 looks very promising. We've looked across multiple populations and it seems to be exceedingly rare. That would be kept for further attention. The reason you need these population databases of variants from different ancestral groups is for a situation like this. If we look at variant 3 and we only look in population 1, we think it's of high priority. It looks promising. It's very rare. But all we needed to do was look at other populations and see it's quite common, and we should throw that out. Similarly, variant 4 is one where it seems in a few populations to be quite rare. We could be misled, but by having a good data resource about population 4, we would find it actually can be very common in some populations. We should throw that one out. This is why we are developing databases of variants from different ancestral groups, from different populations, for exactly this filtering step, so we can get this down as small as possible. I don't have time to go through it, but there is a growing literature that shows that the more and more you have better and better population data about variants, you decrease incorrect genomic variant classifications. And meanwhile, you also, as you can see on the right, you increase your diagnoses by having more and by having study recruitment and developing these data resources for more diverse groups. And in doing so, you, at the end of the day, improve your clinical testing. And so first you detect the variants Then you filter and you prioritize the variants, and then you have a modest number that you can analyze to see if they're medically relevant. For this, you need knowledge bases. And for this, we are establishing the clinical relevance of prioritized genomic variants through knowledge bases that are increasingly being developed across the world. The prototype for this is a one that we support at our institute called the Clinical Genome Resource, or ClinGen. Its mission is to build and support an authoritative central resource that defines the clinical relevance of genes and variants for use in precision medicine and research. It involves an international group. We now have a global network of people, perhaps some people who are listening to this talk, from 69 countries and 2,700 experts from around the world who help curate these limited variants to try to figure out which of the ones play a role in human disease. And I can just show you some numbers about how many correlations we have between diseases and genes and variant pathogenicity curations and actual actionable, clinically actionable recommendations based on specific variants. And so what I hope you've appreciated by this grand challenge I told you about is that in order to, what is required for accurate and equitable analyses of a person's genome sequence is an appropriate matched human reference sequence, or eventually in the future, a human pangenome reference, population databases with aggregated genomic variant information, such as NOMAD as one example, and also robust knowledge bases of curated information developed by expert panels, such as ClinGen. And all of these things are critically important in order to sort of take full advantage of what we're trying to accomplish in making genome sequencing part of medicine in a fair and equitable way. So that's what I wanted to tell you, but I wanted to just sort of end by a couple of things. We are working very hard in the genomics community at pushing forward everything that's going on in genomics, especially human genomics. I described to you some of these medical applications. In fact, we encapsulate this under our mantra for our institute called the Forefront of Genomics, which very much reflects this 2020 strategic vision that we published that I alluded to earlier on, that explicitly talks about improving human health at the forefront of genomics. I would really encourage, if you haven't read these 10 pages in Nature, you should do so. It very much captures many of the things I talked about in my talk today. And there's a URL, you could download this for free, and there's lots of information about our strategic vision. I thought I would just sort of end, though, on a very upbeat note by telling you that the last part of that strategic vision that we published included some bold predictions for what we thought was going to happen this decade. And we put that in the Nature paper right here in this box. And here I'll blow up the box where we put out 10 bold predictions. And in those 10 bold predictions, we specifically talked about them in the paper. And we said, but now more than ever, solutions to these problems seem to be within striking distance. And towards that end, and with the characteristic spirit of genomics audacity, we offer 10 bold predictions of what might be realized in human genomics by 2030. And we pointed to the box. But we sort of found that those two sentences were not adequate to really convey the excitement of these bold predictions. And they are bold. So let me share these with you in a short video. Uh-oh. I worry you don't hear the audio on this. Are you able to hear that?
No, Dr. Green. Yeah, sorry, but
Yeah.
yeah, I'm sorry. Can we let it go like this if that is fine with you?
We can.
I'm sorry.
It's just that you really need to hear it. So maybe I'll skip this. I will just tell you. Maybe I'm going to skip that. What I would say is everybody should go to YouTube. And I think if you go to YouTube and you put in NHGRI 10 bold predictions, that video will come up. And there's something with the Zoom with the audio. So let's skip that. And we started a little late anyway. So let me just say, please go to YouTube and see that. It's about a 4-minute video. It's a lot of fun. So that was what I was going to say. And basically, I was going to end just by pointing out that what I've described to you is basically a 21-year pivot. We went 21 years ago to completing the Genome Project to now pursuing genomic medicine. And what's wonderful is it's now in focus. And while I think there's a lot more still to come, we're going to see tremendous advances in genomic medicine over the next 10 to 20 years. I think the clarity of these initial examples have been quite gratifying because it really has justified why we pursued the sequencing of the human genome as part of the Human Genome Project. And so I'm going to stop there, except to say, please follow me if you want. There's a lot of ways to stay connected. I put out a monthly newsletter. I'm on social media in various channels. And there's a wonderful landing page if you just go to genome.gov/stay connected. That'll describe all the ways to stay connected with me and with my institute. And so I will stop there. And I'm happy to take any questions you have. I don't know if we're going to do them live or we're going to do them through chat. So I will stop sharing and see if I can see people. There we go.
Thank you. Thank you so much, Dr. Green, for this scintillating talk. We are quite honored to have you today. There are a few questions. If I can read it for you, would you mind
Sure.
answering them? Yeah, please.
Oh, happy to.
Yeah, sure. So, Jay Kishan asks a question, how India should pursue personalized medicine as world's largest, most diverse cohort.
How your country should pursue precision medicine?
Yeah. Please.
No, I think it's a great question, and I certainly, part of the reason I wanted to show you the data about pangenomes and reference genome sequences was to really stress the point that to be able to take advantage of all these advances in genomics, you really need to have genomic data from your population, from your ancestral groups. And I would certainly encourage all parts of the world, all countries, to try to generate those kinds of data and those kinds of reference sequences, in addition to having these lots of collections of genomic variants, because they're going to be critically important for being able to do the kinds of diagnostics that are going to be important. So I would say I don't really know all the things happening in India right now, but you want to have data from your populations in order to move this forward.
Yeah, we recently finished 10,000 genome sequencing project, and that was just published. Thank you so much, Dr. Green. So we have Amit Rawat asking, is there a concerted effort to include individuals with different ethnicities for reference genome?
Yes, that's exactly what the Human Genome Reference Program is all about. That's the program we're collecting 500 people, or it's five so far, and it's from all over the world. We don't need Northern Europeans, or we don't need Europeans anymore. We need from all over the world. So our reference program is exactly that, capturing diversity, ancestral diversity from all across the world.
Yeah, on that front, he asks, whether or not AI-based tools, could probably, be illustrated, to analyze these human exomes or human genome, sequences.
100%. We are very excited about AI. There's several applications that are incredibly exciting. The human genome sequence is a really hard thing to read. We barely understand the genes, and yet the non-coding regions that are functionally important, that's a language that the human eye has a lot of trouble with. So AI has played a major role in developing models and so forth to understand not only genome function, but the role that variation plays in influencing genome function. I think another area, because the previous question was about precision medicine, which to me is about bringing together genomic data and other data about people, including their medical data, including other omic data. To me, AI is perfect for that, because when you're starting to integrate disparate data types, having AI as a fundamental tool is going to be very impactful. So there's significant amounts of research. In fact, I'm very gratified to see some of the AI world experts gravitating to genomics because they see some of the opportunities because of the size of our data sets.
Wonderful. Thank you. There was another question from Rahul Mishra. Since there are a good number of variants of uncertain significance, popping up in the databases, is it ethical to share the genomic information with families concerning the privacy and probably the discrimination? Yeah.
Yeah, I agree with you. There's lots of variants of uncertain significance. Sharing them across the world sometimes is very helpful, because we routinely see situations where we're not sure if a variant is causative for a rare disease. And then by sharing this information, you find another patient with the same rare disease at the other end of the world with the same or a similar variant. And sometimes that information actually is incredibly powerful for demonstrating that indeed that variant of uncertain significance is significant. So part of the reason for getting the sharing of data and connecting patients from all around the world is to get some validation that an uncertain variant turns out to be certain.
Yeah, thank you, Dr. Green. Can you please take a few more questions, or
Sure.
are you running out of time? Yeah, yeah. Thank
No,
you.
we're good. I can take a few more.
Oh, thank you. Yeah. So, Ayusha Agarwal, she asks, is it possible yet to completely cure a genetic disease in adult humans rather than just delay with drugs or stem cell therapies?
Well, I think there's a lot of excitement around a couple of areas where you can cure genetic diseases. I mean, for a long time, sickle cell and the sickle cell mutation was discovered even before the Human Genome Project. The Genome Project did nothing for sickle cell disease, essentially for the most part. But now, through CRISPR, there are clinical trials going on that are successfully curing sickle cell through CRISPR-based methods and bone marrow transplants. That's remarkable. I would also similarly say there's a lot of exciting things going on with certain types of eye diseases where again, through CRISPR-based methods, they're curing things like macular degeneration and so forth. So I do think there's some cures that are truly remarkable that are coming out, especially with these tools of genome editing.
Right. On that front, Sherin Sylvia, she asked whether or not it is boon or bane, keeping in view of ethics associated with this. I mean, can we still edit certain genes in lieu of the genomic medicine? Yeah.
So the ethics of genome editing, is that sort of the question?
Yeah. Yeah. Please. Yeah. Yeah.
Yeah. So I think as long as we're dealing with somatic cells, not germline cells, I think scientists, we have to be careful. We need all the right studies to be done, but I think people believe that they're showing the safety of this. They're doing this in a very systematic way for somatic cell editing. Now, of course, germline editing is not regarded as acceptable. It's happened. The scientific community had significant criticism of it happening. We should not be doing germline editing because we don't know what the consequences are for inherited cells having been edited. And so I think we always need to recognize the difference between germline editing and somatic editing. But I think in the case of somatic editing, people believe, there's a risk associated, but the risk is pretty small considering the disease that you are curing. But of course, this needs to all be validated through appropriate clinical research studies.
Thank you, Dr. Green. Just last couple of questions. So,
Sure.
Prashant Ranjan asks, how does multi-omic approaches provide insights into, deciphering these non-coding regions, in the genome? Yeah.
Yeah, it's one of the areas we're very excited about. We just recently launched a new consortium called the Multi-Omics of Human Disease, or MAUD, program. We love genomics, but there are other omics that bring in other data sets, whether it's transcriptomics and, epigenomics and other affiliated data sets integrated with genomic data, we believe will provide a very important new insights about human disease and human health. And we certainly believe that a lot of the mystery of non-coding DNA will be aided by studying RNA, studying epigenomics, and so forth. So absolutely, we are seeing an expansion of omics well beyond genomics. And it should make computational scientists very excited because there's going to be so many opportunities for the integration of large data sets.
Thank you, Dr. Green. I'll probably ask a question myself, if I'm allowed
Sure.
to. Yeah.
Yeah.
I was just wondering, there has been a steadfast focus on the low pass sequencing these days, traditionally moving from 0.75x to 1.5x depth of coverage, or to a lesser extent. At this forefront, there is also a credibility question, that's going on, with respect to prioritizing the genetic variants. So, do you see this as an immense challenge, in the near future?
Yeah, to be honest with you, I think that's highly variable in terms of what the level of coverage is that people get. I mean, I think as part of diagnostics, at least here in the US, they're not doing zero point 0.7 or 1.5. They're doing like 30x coverage. So, I realize this is all cost related, but I don't think I think the difference may be whether you're just doing light survey to detect variants or whether you're actually trying to use this as part of a high accuracy diagnostic or research study. So I think it all depends on the application. But if you're skimming that sequence, that's insufficient for making accurate clinical diagnostics.
Wonderful. Yeah. With this note, I would like to thank you, Dr. Green, for your gracious time. It's been a
Okay,
kind of an incredible experience and a great kickstart to this AdNet Index conference. I would like to
great.
thank you wholeheartedly. In fact, Susan has been very kind enough, in seeking an appointment for you for this wonderful keynote of yours. We Indians keep following nih.gov video casts. We are a great fan of yours. We have been
Great.
following all the talks, and we have a robust bioinformatics society in the form of BioClues and DNA fingerprinting technologies in the form of AdNet, through which you have given this wonderful keynote. So,
Great.
on this note, I would like to thank you again and Susan and the entire team NIH, and of course our
And,
local organizing committee headed by Gyaneshwar Chobe, who is the chairperson for this upcoming conference that's going to be started day after tomorrow, and all the other local organizing committee members like Rahul, Prajwal, and Shailesh, and everyone. Thank you so much,
okay.
Dr.
And by
Green.
the way,
Yeah.
you mentioned NIH.gov. I would also tell you that go to YouTube and go to Genome TV because we have our institute has a Genome TV channel of YouTube. That's where like the video I couldn't show you, but you'll find it. But you'll find hundreds of videos there. So if you really want to hone in on genomics, go to Genome TV channel of YouTube.
We have
And we
been
love sharing it with the
subscribing to
world.
this. Yeah. Yeah. Thank you.
Okay.
Thank you
All
so
right.
much.
Good to see you. Take care,
So,
everyone. Bye bye.
so friends, I hope you'll agree that we had a scintillating talk. So let's give a thumping round of applause to Dr. Green. Thank you. Thank you so
Thank
much.
you very much. Thank you.
Thank you, Dr. Green. Yeah.